Okay, I have 11:30 on my clock, so I think we're going to go ahead and get started. Welcome to today's webinar. This is Using Research-Based Solutions for Integrated Management of White Mold in Soybeans by Damon Smith. I'm Lene Jess, the co-director of the North Central IPM Center, and we are one of 4 regional IPM centers that are funded by USDA NIFA, the Crop Protection Pest Management Program, regional coordination part of that. We have a mission to coordinate IPM across regions, and we started this series because there is a requirement for PDs to give talks on their grant proposals, their research projects, and because of COVID, that wasn't possible last year, so we started this webinar series. Damon has asked that we have put your questions in the Q&A, and then he will answer those questions at the end of the seminar. And so today, like I said, our speaker is Damon Smith. He is an associate professor and field crops extension pathologist, faculty director of the Nutrient and Pest Management Program in the Department of Plant Pathology at the University of Wisconsin-Madison. I'll be putting some links to some of his work, the Crop Protection Network, his website, and then a pertinent video in the chat, so you may see that come up. And again, just to remind you, if you have questions, please type those in the Q&A as we go along. With that, welcome, Damon, and thank you for being part of our webinar series.
All right, thank you, Lene. I appreciate the introduction. Given the time today, I'm going to kind of jump right in here and just get started. And I do want to, some of you may be familiar with white mold on soybean, but I do want to just take a little bit of time here just to introduce the disease and the disease cycle, because it is going to become important as we, go through the presentation. So we have, basically we'll start here with Apothecia development. These are little mushrooms that have an inverted cup. And on the cup here, we have basically these microscopic spores that give rise or are aerially dispersed up into the canopy of soybeans, specifically to senescing flowers, okay? Now this seems like a fairly simple process for the germination of these apothecia, but this is actually quite an intricate process. There's a lot of biology involved here. These apothecia are actually born on these small sclerotia that are sort of like seeds of plants. And these sclerotia are found in the upper 2-inch profile of the soil. And when conditions are right, and the conditions I have listed just generally over here on the left-hand side of the screen, temperature is extremely important for this whole process. So we need average temperatures that are at or below 70 degrees. And really what's happening here is it's the nighttime temperatures that really, drive a lot of this. So these cool nights, warm days, we get dew and high humidity. Those things are very conducive. But there's also a third piece here, which is very important, and that is between row closure. And I'll talk a little more about what we're doing with row spacing and stuff here in a little bit, but with some work that we did with Marty Chilvers at Michigan State University, we know that the between row closure requirement for the development of these little mushrooms is pretty important. We have to have 40% or more between row closure. And so you can actually delay the development by actually spacing soybeans out further, okay? So this whole process is very intricate, lots of biology, lots of environmental interactions that take place here. Then we have the biology of the plant, flowering duration time, very important, drives a lot of the disease levels here. So the longer these plants flower, the longer the opportunity for the fungus to infect the plants. The shorter that bloom duration, the less time that fungus has to infect. Once the fungus infects through the flowers, it then colonizes the stem, and we get these bleached lesions. And it takes about 2 to 3 weeks after infection to actually get to this bleach lesion phase, where we actually see the white fluff and those lesions, and the plant starts to die. And initially, when I got to Wisconsin, there was a lot of folks just trying to apply fungicides once they saw the damage. And we quickly realized that, that wasn't a great philosophy, because basically you already missed the infection process, and our fungicides really are protectants. So the opportunity for the fungus is way back here at the flowering time, and that's the opportunity that we have in season with our fungicide applications. And then once the plants die, we get the formation of these sclerotia, and we've completed the cycle once we run our combines through the field. One interesting thing to note, in Wisconsin, we've also seen a lot of contamination of combines moving from field to field, and that's been a major way of moving actually sclerotia from one field to the next. So that's something to bear in mind too. We do have what's called a monocyclic cycle, so we have only one cycle per season. So all the active inoculum comes from the prior seasons. All right, in terms of how damaging white mold can actually be on soybeans, this is sort of a visual representation. This was a production field in 2014. This was a graduate student I had at the time, Jamie Wilbers, now an assistant professor at Michigan State. And Jamie did a lot of work trying to understand biology and yield loss in the soybean system. And you can see in late August, quite a bit of damage over here in the left-hand side, all these bleached stems, that's all white mold. And then you can see all the premature defoliation and things going on here mid-September. Same field, just a little different perspective there. And you can see pretty significant damage. This field, we were at 50% to 60% yield loss in this particular field. So can be very significant. We also know that yield loss can be variable, and it's variable depending on where the fungus infects the plants. So one thing I want to mention is we use what's called a disease severity index score in our research to actually score damage and try to understand yield loss. And I'll explain why this is important here in a second, but I just want you to understand how we're actually scoring here so that some of this next conversation makes a bit of sense. We do two types of ratings. We do incidence, which is the percent symptomatic plants per plot. And then we also do severity scores. The severity score is a 0 to 3 scale. 0 being no infection, 1 is an infection on a lateral branch, and then 2 is an infection on a main stem, but may not necessarily kill that main stem. And then 3 is pretty much complete death of the plants, or something like you'll see over here in this picture. We then take the two scores, we multiply them together and divide by 3, which rescales this whole thing on a 0 to 100% basis. So 0 be no disease, 100% be complete loss, basically in that field. Now, the reason why it's important to score this way is because the original yield loss curves that were built only looked at disease incidence, and it was a nice linear process where you had a nice straight line where we looked at the increase in disease incidence and yield loss, a nice negative slope. What we heard farmers saying though was, I could have a little bit of white mold, run the combine through and not see a lot of yield loss. So we were curious about that, and we started to realize that over time, breeders have added more lateral branches to the plants. Okay, so it's really important to understand whether we have primarily lateral branch infections affecting yield, or is it main stem infections. And we can see this is actually some work that we published in 2019 looking at yield loss relative to this disease index score. And what's basically going on here is we have a nonlinear relationship when we use this disease index. And what's happening is this is a negative slope up here in the flat part of the curve, but it's primarily where we have lateral branch hits. Okay, and so some resistance, we've added branches over time on these plants, and that lateral branch damage is not as influential on yield reduction as would be a main stem hit. And so once we get into these main stem infections where we have basically little resistance in these plants, lots of damage, that's where we're in this very steep part of the curve. And this cutoff's right around this 40% on the disease index curve. So we have what we call our low pressure disease situations, and then our high pressure disease situations over here on the right-hand side. And you can see quite a significant yield reduction there. In terms of management is a function of multiple things. So we really, we're going to bring a triangle in here. We are pathologists. We like to talk about the disease triangle. This is our white mold management triangle. And so we really have to layer multiple strategies together. There's no one strategy that gives us complete management. And so we're looking at things like field history, mapping the field, trying to understand where there's high and low levels of white mold in a particular field. So maybe we could take advantage of prescription seeding technology and those sorts of things. We're also interested in using field history so we can make variety selection, trying to look at resistance and making the best decision on trying to choose the best partial resistance. Canopy row width, planting populations, crop rotation, chemical controls and biological controls, et cetera, et cetera. So it is a layered thing, or what we call an integrated pest management situation. Now I'm going to dig in a little bit on kind of where we're at right now. And basically the work that we've been doing over the last 5 to 10 years has been based on a few questions from farmers, and I'm going to dig into the top three that we were really confronted with. And the first is, when should I spray for SSR, and what fungicide should work? And is there genetic resistance to SSR or white mold in these varieties? And what cultural practices should I use for my integrated management plan for Sclerotinia stem rot or white mold? So I'm going to kind of cover these. I'll mention some of the work that we have done and kind of where we're headed here as we keep moving forward. I do want to make it clear that we've published readily in these various areas. So I'll first touch on white mold prediction and using fungicides to manage disease. I should mention that this SSR acronym is Sclerotinia stem rot. So that's another name for white mold. So anytime you see that, just fill in white mold in the blanks there. And you can see we have basically 4 research publications that revolve around the work I'm going to talk about here in brief in the next couple of slides. So there was quite a few questions a few years back on, fungicide efficacy, what are the best programs, what are the best timings, and that sort of thing. And some of the work we did with Jamie was actually looking at some of the top 10 programs at that time. And this work was done starting about 2016 and ended up about 2018, 2019. And you can see we have a various modes of action here, and we broke out our relative yield reductions and in terms of percent yield in a particular field. We broke those out in those low disease pressure and high disease pressure situations. So again, if we're in the flat part of the curve, we're looking at the yellow bars. If we're in the steep part of that yield loss curve, we're in that red bar. And so what was interesting is, really in a low disease pressure situation, most everything gave us some yield advantage over the non-treated. So everything here is relative to the non-treated control. So you don't see the non-treated control bar here. So everything gives us a positive here, with the exception of an active ingredient called lactofen, which is actually a product. It's an herbicide called Cobra. Cobra can be applied as a suppression for white mold to soybean. It's a 6-ounce rate at the R1 growth stage. And it's interesting, in the low disease pressure situations, we actually lose 1 to 2% on average across our work. Okay, so here's a case where we have to account for that yield history. We have to look at that disease pressure. We have to understand, are we in a low disease pressure situation or high disease pressure situation, so we make the correct choice. If we went after any of the other fungicide programs here, basically nothing statistically different from each other when we're looking at the yellow bars. It's only in the high disease pressure situation that we do start to see some positive advantages to using products like Cobra, but we clearly see there are some significantly higher performing products here. This is actually, the best one over here is actually Endura at 6 ounces, followed by a product called Preaxer at 4 ounces. So that's a 2-spray program. Then we have Endura at 8 ounces by itself, somewhere between the R1 and R3 growth stage, and then Approach, which is the active ingredient picoxystrobin in a 2-spray program here. So these are kind of the top 3 programs. And then again, Cobra looking okay in those high disease pressure situations. Okay, so we definitely have to account for disease level there, look at our history. When we look at timing in terms of disease index reduction, the more negative the bar here in this case, in this particular graph, the better performance we got during that particular timing. And so these are relative growth stages in soybeans. So here's prior to bloom. So the V5 or emerged, 5 emerged leaves. R1 would be the start of bloom, full bloom at R2. We had a couple of 2-spray options here going, R1, which would be the start of bloom, or the start of pod at R3. And you can see the start of pod and then later pod set here. And you can see there's clearly kind of a sweet spot during that bloom period. And I talked about that in the disease cycle, that we really have to apply our products at that bloom time. And you can see it here in terms of optimizing performance. If we go too early, we don't get as good a performance. We go too late, not as good a performance. So it's somewhere right in that bloom time. And of course, we cover the whole bloom period with a 2-spray program. If the economics allow for that, then we get a nice optimal protection situation. But not in all years does the economics work out for that to happen. So we've developed one app called Sporebuster. This is actually a fungicide calculator app, where we can actually go in and we can run different scenarios based on some economic models that we generated with Paul Mitchell here at the University of Wisconsin. And so basically what you can do, download this for free from either the iPhone or Android store. You can put in your expected price per bushel. You can put in your expected yield for a particular field. We ask that disease pressure situation here because we do change our average net gains and probabilities based on that disease pressure. If you click on the little eye here, you'll actually get a pop-up that gives you a visual of how bad the disease would need to be to fit those different categories. And then the list actually auto-populates to give you the average net gain and break-even probabilities, et cetera. And you can even go as far as getting local costs for treatments. We do auto-populate based on some list pricing, but you can actually get local pricing for these programs and then run those scenarios accordingly. And then this list will auto-populate. So basically you can run those scenarios, try to figure out, optimal programs for your particular field and situation. We continue to work on fungicide programs. This is some data from both Wisconsin and Michigan. So Marty and I put out some uniform trials along with Darren Mueller at Iowa State last year. This is just the Wisconsin and Michigan data because these particular fields had high disease pressure here. Endura continues to be really the optimal program here. Inside the red box here, we actually have the best programs, or what I call the A group. The blue box is the B group, and the green box is the C group, or things not different from the non-treated control. We do tend to try to get creative with different application strategies, trying to T-band things, drop nozzles under the canopy, trying to put things in furrow at plant. And you can see a lot of that creative stuff didn't work very well last year, but you can see some things here did do very well. We have a 2-spray 8-ounce Endura program here that we would not recommend because of the economics of this. That would be a very expensive program, but this we call our standard positive check. And we had a couple of things, especially our Endura Preaxer program, still doing just as good as that positive check. And then we're actually looking at some research treatments as well. So Endura continues to be good. Cobra, we still lose a little yield just from the damage that we get from that herbicide application, but it is a pretty good, cheap option. And again, in those high disease pressure situations might be a good fit. Now, I'd like to transition from product into how do we make that decision on whether we should really pull the trigger or not. And a lot of that decision that should be made about whether we should spray or not spray in a particular season is based on the development of that mushroom. What we've tried to do is really capture the biology of this whole system. If we don't have the mushroom, the little apothecia, which you can see here in the right-hand side of the picture, if we don't have that apothecial form, we don't get spore dispersal. So, most of the infections in a particular soybean field come from within that particular field. Okay, so if we have a history of white mold, we know the inoculum's there. It really comes down to, is that weather such that we're going to get the development of the mushroom and then the subsequent dispersal of those ascospores. So these apothecial, the apothecia are very important in this whole system. And the production of those apothecia is a must in order to have spore flights, okay? Just to show you some data to back that statement up, here's some trapping data that we did and some count data. We had some gridded plots years back where we actually went in, set grids out in fields, and then looked at the number of apothecia multiple times per week, while also trapping for ascospores in those same plots. And you can see we've got different growth stages here. So as that crop canopy gets closer together, we get bloom. So we're starting at the beginning of bloom here, full bloom. And then here's the beginning of pod. You can see we get a nice increase in the number of apothecia over time in those plots, and then a subsequent increase in the average number of ascospores, or spores that we catch. And you can see as the number of apothecia start to decline, we get a nice corresponding response in that the ascospores, we don't catch ascospores on those traps again. And so what we try to do is understand that biology. What are the weather parameters that really, influence the development of that particular mushroom? There have been other white mold prediction models out there. What they're trying to do is actually trying to predict the development of the disease, say a month down the road. What we wanted to do was actually predict the development of the mushroom, because if we could catch that and that coincided with bloom time, then we have really good confidence that we should be putting a fungicide application on. So we did quite a bit of work. I'm not going to take you through all the research and statistics and things. There are 3 publications that back this work up. But what we did is we developed an app, which is actually a disease prediction tool that uses GPS reference weather information to actually provide the risk of whether those mushrooms are going to develop or not. This is, again, a free app that can be downloaded for both the iPhone or Android platforms. As of 2021, we were updated on over 3,000 devices. So just this summer, and right now we're kind of in the peak of our period, and we're running about 500 to 600 forecasts a day, actually. So it's a well-used app, considering that white mold is kind of a north central issue. But it is very nice because you can get basically an at-field prediction. So these are pretty prescriptive. You feed in a little bit of information about the crop stage. So are things blooming? Have we met that row closure? We give you some pictures of that row closure. And then once you answer some of that information and set the GPS coordinates, the phone does the rest of the work. It goes up into the cloud, grabs the weather information for that GPS reference point, and then runs all the math to give you a risk prediction. And you can run this every day to keep track of what's going on. So it's a nice tool. We have had a lot of interest in this tool and clearly a lot of use there to kind of make that decision. Just to show you kind of a real world situation where this type of technology could be used. This is a strip trial in Marathon County a few years back, where we actually looked at the timing of application of the approach program. So the typical approach picoxastrobin program here would be a 2-spray program. So you can see 9 ounces of approach out here on this side of the field. And then we've got the same program out here on this side. The farmer yielded these. And so these are averages. These are not replicated. These are just straight strips through the field, but you can get an idea, how effective at that time these 2-spray programs were. Here's the non-treated strip. So clearly adding fungicide here at the right time could be beneficial. And then you can see the R1 program. So approach R1 by itself, not very effective, a little bit of a reduction. But R3 application, so the single application by itself, very effective, and in fact, just as good as the 2-pass and even a 3-pass. So the farmer just wanted to say, all right, if I just got really crazy, would I be able to preserve any more yield here? And you can see probably not statistically different, only about 2 bushels, which wouldn't pay for that 3rd application, because you can see a lot of the work done at that R3 timing. If we go into the Sporecaster app and we actually graph the probabilities of apothecial development, or we can think of this as risk over time, you can see that, he was kind of in high risk all through the bloom time. So we've got the start of bloom right here on the 14th of July. But you can see as he moves towards R3, we start to really rise again, and then things start to drop again. So this is the area that we'd be very interested in utilizing Sporecaster. And you can see if we optimize that timing, we can really get the maximum benefits out of these particular products. So we see farmers using this technology 2 ways. One is to just ask the question, should I spray or not? And then the other way is, a lot of farmers know they're going to apply fungicide in the north central for white mold control. They just have to. They have high pressure year in and year out in these particular fields, but they really want to optimize the applications of these products and get the best benefits. And so they're using the forecasting tool to make that optimal decision. And I'd like to just transition a bit into resistance and what we know about resistance here. Again, we've published extensively on resistance in soybean. What I can say based on a lot of this work is we're still sitting solidly in an area of high partial resistance, but nothing complete. Here's a test that we ran in the greenhouse a couple of years back. We have a greenhouse assay that we use, which I'll talk about here briefly. And you can see these were actually some of the best products, or I'm sorry, the best varieties out there at the time in terms of field resistance. But you can see once we actually put the fungus on there, there's very little what we call physiological resistance. So the fungus or the soybean plant doesn't actively fight the infection. Okay, and the way we do these is we use a pretty simple process. We actually use these inverted pipette tips. We punch little plugs out, and we affix the plug on a cup petiole on the lateral or on the main stem of a plant. And then we simply measure the length of this lesion, and we can track this over time, because not only is there a level of resistance at limiting the initial infection, but we also see limiting the expansion of that infection over time. So our partial resistance actually slows down that infection process or eventually stops it if the plant can recognize early enough that it is infected. Another layer to this complicating scenario here is that we actually have, differential levels of interplay of the fungus and the plants. Different isolates give different responses. So you pick one plant up in one area and you drop it in another area, and it might have looked resistant in one area and would be susceptible in another. So we've published extensively on the variations in isolates. And we also know that various varieties respond differently. So this is some of our latest work. We're actually trying to pull out a set of check lines so that we can then score commercial varieties and then bin them in resistant, moderately resistant, or susceptible. So something like Dwight here would, if things look sort of similar in terms of lesion length increase here, then we'd bin that as susceptible. Something that looks similar to this line we call 5282B here in the yellow, that would be resistant, and the things in the middle would be sort of moderately resistant or susceptible. And you can see a lot of interplay again with our different isolate numbers here. What we're doing is not only utilizing that kind of information to inform farmers on some of the commercial varieties, but we're also looking at that check panel and trying to understand resistance levels and how those interplay in our forecasting system. So we're accurate about 80% of the time with Sporecaster, and we believe that we can increase that accuracy if we could actually differentially spray based on the resistance level and those various lines. So this is just some work that's just getting started with a graduate student of mine, Wade Webster. He's actually deployed these various check lines and he's spraying them at different action levels or different risk levels that we call low, medium, or high. And you can see these varieties tend to respond differentially, with Dwight highly susceptible here, really requiring a low threshold for application. But some things like SSR 5170 here actually may not need to be sprayed at all. So we're actually seeing some breeding lines being highly resistant, may not even need fungicide application because that fungicide really gives us no incremental increase. So we're going to be continuing forward in this work and trying to adjust Sporecaster accordingly. I'd like to take the last few minutes here just to kind of show you where we're headed in terms of integrated management plans. We did receive a USDA CPPM award in 2018. We're finishing up our third year of work here. We're actually in the final stages of draft for a research publication that's going to be submitted here pretty soon to the Journal of Agronomy, actually looking at this in integrated management practice, where we actually looked at layering planting populations on top of row spacings and then layering fungicide applications on top of that. So really trying to dig in and put all the different pieces of the puzzle together to try to understand what gives us the biggest incremental reductions, and where should we be putting our eggs, if you will, into our basket here. Lots of different locations across the upper Midwest here. I'll cook down a lot of information into just one slide here, which I'll take just a little bit of time here to kind of break this out for you. When it came to disease severity index scores, we have our narrow row spacings, our 15-inch row spacing over here on the left-hand side. We've got the wider row spacing, a 30-inch row spacing over here on the right-hand side over here. And you can see the spray programs where we either didn't treat or we used a model-based application of the fungicide approach or a standard two-pass application of approach. You can see the standard did pretty well. Model not quite keeping up, but clearly there's a pretty good reduction where we go into those wider row spacings. The issue with the wide row spacings, though, and farmers know this, is that when we go out to a 30-inch row spacing, you can see there's a pretty good decline in yield. And you can see it in this lower panel here, where we get a pretty good loss in yield here by widening row spacing. So they're not really inclined to go to those wider row spacings unless a field is just really, really infested with a white mold fungus. That's a case where they might do that. So what we're seeing a lot more of is actually sticking with the 15s, but they're dropping planting population. And our work sort of corroborated what the agronomists have been showing, in that, somewhere in that 140,000, 120 to 140,000 range, this would be 110 and 140,000, we start to get our economic optimal planting population. And that economic optimal also helps reduce white mold and gives us a yield that's comparable economically to dropping, 170 or even 200,000. So we're seeing a lot of farmers here in the Midwest now dropping populations, not only to be optimizing their economics, but trying to reduce that white mold situation. And so if we get that right and we can pair that up with a particular moderately resistant variety, we can almost eliminate the fungicide program. At least that's some of the work that we saw here as well. We've since pushed forward a little bit here. We're now asking the questions, okay, if we do a high and a low seeding population, we layer a fungicide on top, and we maybe have a manured field. So we actually have high nitrogen, 150 pounds of nitrogen applied to that plant to duplicate a manured field here in the right-hand panel. And then on the left-hand side, no nitrogen a plant. You can see that we get some nice reductions where we keep those planting populations down in that 120,000 range compared to 160,000. And if we layer a really good fungicide like Endura on top of that, we can really optimize the reductions of white mold. And you can see the corresponding yields as well, where we get that just right. Clearly here, where we have a really high performing fungicide, even in the high nitrogen situation, we can protect those high seeding rates. But if we want to try to manage this and reduce the return of inoculum back to the soil, we'd probably want to be in a situation like this over here in the left, where we're dropping 100,000 seeds, not applying nitrogen to keep that canopy less lush, and putting a fungicide on, maybe with our Sporecaster tool. Just to finish up, I'll briefly cover where we're headed now. In addition to what we've been doing with row spacing and planting populations, there's a lot of interest in cover crops and roller crimping. We've got some work out this year on farm. We got here's actually some of our on farm work. We found out that a roller crimper can actually be an excellent picnic table for a lunch. You can see some of our plots here in the back that are on farm, and you can actually see the roller crimper in play here actually. And the idea here is that if we plant a rye crop, we let it flower, we can come in with this particular tool, we can crimp or break that main stem at the flowering time. That'll kill that rye crop, and that rye crop then makes a nice thick mat on the surface of the soil. And we can utilize that thick mat not only for weed control, but also white mold control. And so there's been some really nice work. Sarah Pethiebridge at Cornell did a nice study where they actually looked at rye residue and the effect that it has not only on the incidence of white mold, but also the development of the mushroom. And I just want to bring your attention actually over here to the far right, where they looked at soybeans, where they had the rye residue and no rye residue basically. And what happens is not only does that thick mat form a physical barrier for reducing spore flights, but it also reduces the full production of the cup on top of the apothecia. Okay, and so basically what's happening is it's stopping that biology, it's stopping the production of the mushroom, and without the mushroom, we don't get infection. And you can see a corresponding significant reduction in white mold if we go back here, over here to the left, where we have that rye residue. We've done some work in my lab trying to understand what's going on there in terms of the biology, and there has been some work published on this over in Europe. And basically what's happening is there's a change not only in the amount of light that penetrates that thick mat, but there's a change in actually the wavelengths of light. It's interesting that there's actually a really crucial wavelength between 300 and about 320 nanometers that has to be supplied for the full formation. And so the thought process is that by adding that mat, we're changing the light scenario such that the mushrooms can't even form at all underneath that mat. So a lot of work being done here. We have some research plots this year, as well as some on-farm work trying to follow up on that. So just to conclude, this is kind of my take home, and I can actually kind of cook this all down in 3 major points, where it's basically adjusting cultural practices, maybe layering a fungicide program on top. Once you get that row spacing right and the populations right and the varieties right, maybe looking at that fungicide program. But we do know those fungicides are just sort of a I call them, fungicides only make a bad situation less bad, or they're a band-aid, right? And so there's a heavy reliance on those, but we can really do a lot of good actually with a lot of the cultural practices here. If you are going to go the fungicide route, we're pushing those prediction tools. It's better than nothing. At least you have some informed decision there, and then making some decisions on the economics of some of the products that could be used for your particular farm. So I'd just like to acknowledge there's of course a whole bunch of people and funding sources here. There's lots of folks in my lab and some companion labs here at the University of Wisconsin. We also have quite a few collaborators we've worked with, as well as folks at different universities and things. We've got a lot of funding agencies over the years, including the USDA, and then also the North Central IPM Center, who's helped us with some of that work as well. So with that, I'd be happy if we have time, Lene, to take some questions. I think I'm only about 5 minutes over, but here's my contact information. If anybody has anything they want to ask me after this, feel free to reach out via my office phone or email, or I'm on Twitter. I'm pretty active there at BadgerCropDoc, and then of course the website BadgerCropDoc.com.
All right, thank you, Damon. That was fascinating. I have a couple questions for you. The first one is white mold pretty much evenly distributed in a field? And then how does that affect treatment?
Yeah, that's a good question. And actually we were, I just teamed up with a ag engineer to actually understand this a little better. So it's widely known that white mold can be very aggregated in a field or in particular hotspots across the field, and there's many reasons for that. One of them, that combine with sclerotia and it rolls in and turns on the first time and then blows all the sclerotia out. And so there's a hot spot, right? So that's just one reason. There's also, in parts of Wisconsin, we have a lot of topography with a lot of rolling hills and things. And as you might expect, areas where heavy humidity sits, that will be a hot spot down in the valleys. And so it's been, known for a long time that you can have certain areas, and it's usually not uniform across the field. And so, there is some opportunity here with precision agriculture to actually sort of fine tune or do prescriptive seeding rates. So I actually teamed up with an ag engineer. We've got a graduate student who's going to be starting this fall. And some of the idea, what we're going to be doing is trying to track or map white mold infestations in the field, and then look at prescription spraying technology and then prescription planting technology to sort of fine tune what happens within the field. So there's certainly some opportunities there to sort of optimize that instead of doing one treatment across the whole field.
Okay, great. So the second question is, what kind of traps are you using for the ascospores, and then where are you placing them in or around the field?
Yeah, that's a good question. Then we went round and round at the time we did this, and I'll admit we started doing a lot of this work back in 2013 and 2014, where some of the automated trap technology was still just coming online. So we actually relied on some of the old literature looking at, there's these bromophenol blue plates. So they look like a little purple plate. An ascospore lands and it produces what's called oxalic acid. That acidic environment will actually change that purple to a yellow color, and then you can count colonies. And so we would trap between the hours of 9 a.m. and 2 p.m. in a field while we were doing our epithelial scouting. We placed transects of these plates across that grid, and then we also had plates around the perimeters of fields, and so we could actually track across the grid and line up that spore trapping. Admittedly, might be nice to go back now with some of the automated trap technology that we have available to us and actually revisit some of that. But we did learn that, most of the inoculum is again coming from within that field. We did catch very little actually out of the perimeter. And so that was good support for, the subsequent work where we actually developed the models.
Great. Next question is, what weather station data does the app use? Is it just the NOAA weather stations, or is it some of the other type weather stations?
No, we're actually using gridded interpolated weather. We started out when we first got into this, we were actually using the Dark Sky API. And so there's lots of weather services out there that aggregate data from the NOAA stations and city stations, and then they have their own algorithms to sort of tie all that stuff together into a meaningful prediction on these, somewhere between 2 and 5 kilometer grids. It varies based on the company. And so we initially started with dark sky. Over the last couple of seasons, we've transitioned now, and we're actually using IBM weather, so the weather company weather, which is what most of the big companies are utilizing anyway. So we tapped into that, and part of that is because Apple bought dark sky, and they're not going to let us actually be able to program for the Android platform. So we want to make sure that we were able to supply the app to both platforms, and so we wanted to have an open source API. But it is all gridded, interpolated weather data. We do use meteorological grade weather stations on an annual basis to double check that weather. But it's 2021, and this interpolated weather is just really good stuff. There's really little need in my mind these days to have in-field weather stations other than corroborate that weather.
Yeah, great. And the last question I have, unless I see another one popping up in the QA, is have you seen any fungicide resistance yet?
Yeah, excellent question. And yes, we actually have started to see, we have some data to suggest there is some resistance starting to build, especially against the approach products of the picoxystrobin. And we believe that that's why that Sporecaster treatment and the integrated work that I showed you quickly here towards the end, where we believe that could be one reason why that particular program didn't work, is we started to see that program slide here in the North Central, and there's some data to suggest that we have resistance building up against picoxystrobin. So we've since pivoted, and you saw in the work that we did last year, we're now utilizing Endura and 1 and 2 pass programs. While an expensive program, there's no known resistance there yet, and so we can better track, the performance of apps like Sporecaster and some of our other work. So, yes, that is something that's on our radar. And Darren Mueller at Iowa State has been pushing us pretty hard. We need to start looking at multiple mode of action programs to try to mix up some of this. And so I think that's where we're going to be headed to on the fungicide testing side of things, is trying to mix these things up to try to slow down this resistance issue.
All right. Well, thank you. I don't see any other questions in the Q&A, so I want to thank you again, Damon, for the presentation. It was very informative. I learned a lot during it, and we thank you for that. And just a reminder to everybody, our next Pests and Progress webinar will be on Wednesday, August 18th, and our speaker for that will be Bob Wright from University of Nebraska at Lincoln, and he'll be talking about his EIP project, highlighting crops, pollinators, housing, and school IPM. If you want to see a list of our upcoming webinars, you can go to our website. It's just ncipmc.org. You can see the webinars that are on there. And then you can also, if you'd like, our central issue is our newsletter that we have. You can sign up for there on the website also. So thank you very much, Damon. I hope everybody has a great day, and hopefully we'll see everybody in August. Thank you.